This lecture is on chemical reactions. What are chemical reactions? Chemical reactions occur whenever chemicals combine with or dissociate from other chemicals. Energy is stored as potential energy in chemical bonds. Energy is released when chemical bonds are broken. In the body, energy is released as electric, mechanical, or heat energy. Reactants are the chemicals involved in the reaction. They are written on the left side of the equation. Products are the chemicals formed from the reaction. They are written on the right side of the equation. This is an example of a chemical reaction. The reactant is adenosine triphosphate, known as ATP. It is a nucleotide molecule containing adenine, a ribose sugar, and 3 phosphates. When the reaction occurs, the products formed are adenosine diphosphate and an inorganic phosphate, and energy is released. Metabolism is the sum of all chemical reactions in the body. This is an example of cellular respiration, a metabolic pathway that occurs in the mitochondria. Carbohydrates may enter the pathway. Lipids may enter the pathway, and so can proteins. Synthesis reactions build molecules and store energy. An example would be A plus B reactants are joined together to form the molecule AB. Synthesis reactions are anabolic. They build molecules such as muscle proteins. These reactions are endergonic, meaning they store energy. In an endergonic reaction, the amount of energy required is very great. Energy must be added for an endergonic reaction, in which reactants are converted to products with a higher energy level. This illustration of a synthesis reaction shows smaller particles are bonded together to form larger, more complex molecules. In this example, amino acids are joined together to form a protein molecule. The amino acid monomers are joined together to form a polypeptide chain, which will end up forming a protein. Decomposition reactions break down molecules and release energy. The example is the molecule A and B, and the reaction breaks it apart to form A and B. Decomposition reactions are catabolic. They break down molecules such as glycogen. These reactions are exergonic, meaning they release energy. As we can see, the amount of energy released is going to cause the products to have a lower energy than the reactants. In this example, decomposition reactions result when bonds are broken in large molecules, resulting in smaller, less complex molecules. In this example, glycogen is broken down to release glucose units. The glucose molecules can then go into a metabolic pathway to form ATP. Exchange reactions occur when atoms or electrons are exchanged between two chemical structures. Exchange reactions has both decomposition and synthesis components. The example is the reactants molecule AB plus C, the reaction will yield A plus molecule BC. These are the most prevalent reactions in the body and include the production of ATP within muscle tissue. This example shows an exchange reaction in which atoms, molecules, ions, or electrons are exchanged between two chemical structures, and it has the same formula. Molecule AB plus C undergoes a reaction to form A plus molecule BC. The image shows the molecule of creatine phosphate and a molecule of adenosine diphosphate. Creatine phosphate would be the AB in this reaction, and ADP would be C. After the reaction has occurred, creatine has lost its phosphate. It is now A, and adenosine diphosphate has gained that phosphate and is now adenosine triphosphate, ATP, illustrated by BC. Oxidation-reduction reactions are a type of exchange reaction that involves the movement of electrons from one chemical to another. The structure that loses an electron is oxidized. The structure that gains an electron is reduced. These reactions always occur together. Electrons may be moved alone or with a hydrogen ion. This is very important in cellular respiration. In this image, molecule A is shown with 2 electrons attached. Molecule A is going to lose those electrons in the process of oxidation. Molecule B will be the molecule that is accepting the electrons, and it will be reduced in this reaction. B will now most likely shuffle the electrons to another molecule somewhere down the metabolic pathway. Glucose oxidation in cellular respiration is the most common example of the redox reactions. The reaction is reactants C6H12O6, which is glucose, plus 6 molecules of oxygen, 6O2, reacts to yield 6 molecules of water, 6H2O, plus 6 molecules of carbon dioxide, 6CO2, and ATP. This is the basis for all reactions in which food molecules are broken down for energy. Glucose oxidation begins in the cytoplasm with glycolysis. Electrons travel with hydrogen ions into the mitochondria. The hydrogen ions form a gradient and flow through ATP synthase enzymes located within mitochondrial membranes. These enzymes join adenosine diphosphate to inorganic phosphate, indicated by ADP for adenosine diphosphate and P subscript I for inorganic phosphate. Fatty acids and amino acids can also be oxidized within the mitochondria for the synthesis of ATP. This illustration shows the hydrogen ion gradient formed from the shuffling of electrons through the cellular respiration metabolic pathway. The hydrogen ion gradient is more concentrated on one side of the mitochondrial membrane. The hydrogen ions will then flow along their concentration gradient through the ATP synthase enzyme. As the hydrogen ion flows through the enzyme, it powers the enzyme to join adenosine diphosphate to an inorganic phosphate. Remember, this is an endergonic reaction, thus it requires energy in order for it to occur. ATP cycling is the continuous formation and breakdown of ATP. ATP is formed when food molecules are oxidized. This gives the energy required for ATP synthase to join ADP to an inorganic phosphate. This is called phosphorylation. ATP is then oxidized to aid endergonic reactions. In ATP oxidation, ATP is hydrolyzed into adenosine diphosphate and an inorganic phosphate. The energy released from ATP hydrolysis provides energy for cellular work. Only a few seconds worth of ATP are present within a cell, and so ATP, ADP, and inorganic phosphate must be continuously cycled. This image shows ATP cycling. We'll start with the molecule of ATP. Remember, it has the nitrogenous base of adenine, a ribose sugar, and 3 phosphate groups. ATP is a nucleotide. In ATP hydrolysis, ATP will be split into adenosine diphosphate and an inorganic phosphate. During ATP hydrolysis, energy is released. This is an exergonic reaction. Adenosine diphosphate must then be rejoined to another inorganic phosphate to form ATP so that it can continue the cycle. It takes energy to join the inorganic phosphate back to an adenosine diphosphate molecule. This is where cellular respiration comes in. It uses energy provided from the oxidation of carbohydrates, proteins, and lipids to supply the energy necessary to join the inorganic phosphate back to a molecule of ADP. And there we return to our ATP molecule, and the cycle can begin again. In an irreversible reaction, there is a net loss of reactants and a net gain in products. An example of an irreversible reaction is glucose oxidation. We can either join molecules together or we can break molecules apart. In glucose oxidation, we're breaking molecules apart, such as glucose, and we'll end up seeing an increase in the amount of products from glucose oxidation, but at the expense of the glucose itself. We will see a decrease in the amount of glucose. This is why we have to continue to eat. So this is an irreversible reaction. In a reversible reaction, the reaction does not only proceed to the right, so we're not only making products. There's going to be no change in the concentration of either the reactants or the products. We're actually going to go towards what's called chemical equilibrium. An example of this is the carbonic acid and bicarbonate ion reaction seen in the blood. This is how the pH of the blood is buffered, if you recall from the lecture on chemistry, the ions and molecules. A reversible reaction is usually indicated where the arrows are going towards both the products and the reactants, indicating that the reactants become the products, and then the products can end up becoming the reactants again. So it just continues to cycle into chemical equilibrium. So an increase in reactants or a decrease in products drives the equation to the right. A decrease in the reactants or an increase in the products drives the equation to the left. Here is the example of the carbonic acid and bicarbonate ion equilibrium in the blood to maintain the pH of 7.35 to 7.45. As we build up our levels of hydrogen ions and bicarbonate ions, our equation will then move to the left. We will form carbonic acid, and it will break into water and carbon dioxide, and we'll breathe the carbon dioxide out. If we have a buildup of our reactants, carbon dioxide and water in our blood, it will form carbonic acid. And then it will form hydrogen and a bicarbonate ion. And the kidneys will either reabsorb or excrete the products as needed. So this reaction is constantly going left to right to maintain the chemical equilibrium of blood pH 7.35 to 7.45. In this example of chemical equilibrium, step 1, the reaction begins. No products are yet formed. There is a high rate of collisions between molecules A and B. The rate of forward reaction is high. In steps 2 and 3, the products are formed. Collisions between reactants decrease, the rate of forward reaction decreases, and as we build up products, the reverse reaction will begin. In step 4, the rate of forward reaction is equal to the rate of reverse reaction. The dynamic equilibrium will be established and our concentrations will remain constant. Again, this is very important in maintaining homeostasis such as blood pH. Now let's talk about activation energy and enzymes. The reaction rate is the measure of how quickly a chemical reaction takes place. This determines how much product is formed per unit of time. The activation energy is symbolized by E subscript A and is the energy required to initiate chemical reactions. This is a primary factor determining the reaction rate. The activation energy can be overcome by increasing the temperature provided energy to break bonds. Significant temperature increases in a cell would denature proteins, however, and thus enzymes are used instead. Enzymes are globular proteins that act as biological catalysts. They accelerate chemical reactions without being changed in the process. They increase the rate of product formation by decreasing activation energy without raising body temperature. In the first illustration, we see that the activation energy required without the enzyme is extremely high. This is the energy that is needed in order to join the reactants together to form our product. With an enzyme, the activation energy is significantly decreased, and it takes less energy to join our reactants into our products. Enzymes are beneficial to our body's homeostasis because they increase the rate of reactions within our body without increasing the temperature. Remember, we must remain at a constant body temperature between 98.6. The amino acids in an enzyme form a unique three-dimensional structure called the active site. Remember, an enzyme is a protein. and therefore it is made up of a chain of amino acids. When an enzyme is catalyzing a reaction, it temporarily forms the enzyme-substrate complex. The substrate is the reactants that are acted on by the enzyme. They become the product. This illustration shows the substrate in red. The enzyme is in blue. The active site is composed of amino acids that will react specifically to this substrate. Enzymes have specificity. They will only act to their particular substrate. When a substrate binds to the active site on the enzyme, it forms the enzyme-substrate complex. And when the enzyme is in the enzyme-substrate complex, the reaction can occur. In this case, a hydrolysis reaction is occurring, where the reactant is broken into smaller molecules. These smaller molecules are the products and are now released from the enzyme. This enzyme is now ready to interact with another substrate. An enzyme's active site exhibits specificity. The enzyme conforms to a specific substrate, enclosing it in an induced fit. This helps the enzyme catalyze only one specific reaction. So for every substrate that we need to work on in the metabolic pathways within the body, we must have the appropriate enzyme. This means that there's going to be a lot of enzymes. Here's the mechanism of action for an enzyme in a synthesis reaction. This synthesis reaction shows glucose molecules being synthesized into a glycogen molecule. Our reactants are glucose monomers. They are the substrate for this particular enzyme. The glucose substrate binds to the enzyme in step 1, forming an enzyme-substrate complex. Remember, when the enzyme-substrate complex is engaged, the reaction can occur. In step 2, the reaction is that the enzyme changes shape, resulting in an induced fit between the substrate and the enzyme. And step 3, a bond is formed between the new glucose molecule and the growing glycogen molecule. In step 4, the product is released. In this case, the product is glycogen. The enzyme is now free to bind to other substrates, but remember, it is only going to bind to glucose, because this particular enzyme is specific to glucose. If we were building amino acid chains, we would need another enzyme. If we were building fatty acid chains, we would need another enzyme. If we were breaking down glycogen into glucose, we would need another enzyme. You see where I'm going. Here's the mechanism of action for an enzyme in a decomposition reaction. In this particular decomposition reaction, lactose is being digested to glucose and galactose. Lactose is different than glycogen. Lactose is milk sugar. It has a glucose monomer, but it also has a galactose monomer. The enzyme that will work on the lactose substrate is the enzyme lactase. So in step 1, the substrate lactose binds to the enzyme, forming an enzyme-substrate complex. Now that the enzyme-substrate complex is formed, a reaction can occur. In step 2, the enzyme changes shape, resulting in an induced fit between substrate and enzyme. And in step 3, the bond is broken between glucose and galactose. The products will be a monomer of glucose and a monomer of galactose. In step 4, these products are released, and the enzyme is free to bind to other substrates. But remember, this enzyme is specific to lactose, and thus will only bind to lactose substrate. Lactose intolerance occurs when an individual lacks the enzyme lactase. Therefore, they cannot digest lactose, which is milk sugar. The milk brand Lactaid has added the lactase enzyme into the milk. And so this reaction is occurring within the carton before it even enters your body, making Lactaid digestible for individuals who are lactose intolerant. Cofactors are molecules or helper ions required to ensure that a reaction occurs. Cofactors are non-protein organic or inorganic structures. Inorganic cofactors attach to the enzyme. An example is zinc. Zinc ion is one of the most important cofactors for enzymes in metabolic pathways. Organic cofactors are called coenzymes. These are vitamins or modified nucleotides. CoQ10 is a popular supplement that acts as a coenzyme in cellular respiration. CoQ10 shuttles electrons between enzyme complexes. We'll see this soon. Here's an illustration of an enzyme that is in the inactive state. It's missing its coenzyme, and it looks like it is a non-protein organic structure, and thus will be called the coenzyme. So that's a big take-home message here. Cofactors are inorganic, and coenzymes are cofactors as well, but they are organic. Remember, inorganic means that they lack carbon and hydrogen. Organic means that they have both carbon and hydrogen. But both coenzymes and the inorganic cofactors are not proteins. So they're going to be minerals such as zinc, or they're going to be vitamins, or they're going to be modified nucleotides. So here we see the protein in its inactive state. It's called an apoenzyme, just FYI. And here we see the cofactor in its non-protein. We know that it's organic, however, because they're calling it a coenzyme. When the coenzyme and the enzyme are bonded together, they call it a holoenzyme, meaning it's together. It is now active. Notice the change in the active site of this enzyme. Without the cofactor, the active site will not fit the substrate. It requires the cofactor, be it inorganic or organic, to fit the substrate. It completes the active site. This makes things more complicated. Not only do you need the appropriate enzymes for metabolic reactions to occur, but you also need the appropriate cofactors. This is why it's important to take your multivitamin and multiminerals every day, because multivitamins and multiminerals will provide a lot of the cofactors that you need for your metabolic reactions. Enzyme names are based on the name of the substrate or the product that they work on or produce. Also, their subclass, which is their method of reaction. If you see the suffix ASE, that indicates that it is an enzyme. Examples: ATP synthase joins ATP to an inorganic phosphate. So here we see that it is named according to its reaction and the products it forms, which is ADP, and inorganic phosphate to create ATP. So it is named after the product that it makes, ATP. And because it joins them together, it's called the synthase. Notice the ASE. DNA polymerase helps to form DNA. Thus, DNA is the product. It's a polymer because it's many nucleotides joined together. So what it's saying in this name is the enzyme, DNA polymerase, builds DNA. Lactase digests lactose. It is named for the substrate that it works on. It's working on the lactose sugar, and it will digest it into galactose and glucose through hydrolysis. So keep an eye out for the suffix ASE, and then when you read the name of the enzyme, it's usually going to indicate the substrates or the products and the method of the reaction. The reaction rate may be accelerated by an increase in the concentration of substrates. A reaction rate will increase only up to the point of saturation. Saturation occurs when so much substrate is present that all enzyme molecules are engaged in that reaction. In this illustration, the rate of reaction increases as the concentration of the substrate increases until we reach enzyme saturation. Once all the enzymes are engaged in enzyme substrate complexes, we are no longer able to increase our reaction rate. The only way that we can do that is to add more enzymes. Enzymes function best at an optimal temperature. This temperature is usually 37 degrees Celsius, which is 98.6 degrees Fahrenheit, body temperature. A moderate fever will result in more efficient enzyme activity, but that's just a moderate fever, very slight. It helps to make the enzymes a little more bendy, and so they can go and attack the pathogens more efficiently. However, a severe increase in temperature will cause denaturation to occur. Remember, denaturation is when the protein is broken into fragments and it no longer can serve its function. In this illustration, as the rate of reaction increases with the increase in temperature up to the optimum temperature. We can go up to 40 degrees Celsius, the slight fever. That is the maximum protein flexibility. And after we have reached that point, any temperature above that will result in enzyme denaturing as weak intramolecular interactions are broken at the higher temperatures, and our rate of reaction will decrease because we're losing our enzymes. Enzymes function best at an optimal pH. Most enzymes function best between a pH of 6 and 8. Changes in hydrogen ion concentrations can disrupt hydrogen bonds, resulting in denaturation. Optimal pH may differ for different enzymes. For example, the enzymes working in the lower pH of the stomach will have an optimal pH of about 2 or lower. This illustration shows the bell curve of pH. So here we see that the optimum pH for most human enzymes is around 6 to 8. So it straddles that neutral range. We can be slightly acidic or slightly alkaline. However, if we get too acidic, the hydrogen ions of the acid will break apart the hydrogen bonds, denaturing our protein. If our pH is too basic, we will lose hydrogen ions from our proteins, and that will also disrupt bonds and cause our protein to denature. Uncontrolled amounts of enzymes would result in an imbalance of substrates and products. Inhibitors will bind to enzymes and regulate their catalysis, which is their rate of chemical reactions. Later, the release of the inhibitor will allow it to function, and the catalysis will continue. Basically, the reactions will continue. Inhibitors can be competitive or non-competitive. Competitive inhibitors bind to the active site. Non-competitive inhibitors bind to a site other than the active site. A competitive inhibitor resembles substrates and binds to the active site of the enzyme. The competitive inhibitor competes for the occupation of the active site. More substrate in the solution will lower the chance that the inhibitor will occupy the active site. Less substrate increases the chance that the inhibitor will occupy the site. In this illustration, the substrate and the competitive inhibitor resemble each other, this teardrop-shaped portion. And therefore, the competitive inhibitor can interact with the amino acids in the active site of this enzyme. So the competitive inhibitor interferes with the active site of the enzyme, so the substrate cannot bind. A non-competitive inhibitor does not resemble the substrate. It binds a site other than the active site. This is called the allosteric site. Non-competitive inhibitors induce conformational changes to the enzyme and its active site. These are called allosteric inhibitors, meaning they are binding to another place rather than the active site. Non-competitive inhibitors are not influenced by the concentration of the substrate. In this illustration, the allosteric inhibitor has bonded to an allosteric site, which is a site other than the active site. The change in the shape of the active site makes this enzyme no longer able to bind to the substrate. Multiple enzymes are usually required to convert the initial substrate to the final product. A metabolic pathway is formed by numerous enzymes. Each enzyme is responsible for catalyzing one step. The product of that step then becomes the substrate for the next step. An example is the electron transport chain. A multi-enzyme complex is a group of enzymes attached by covalent bonds within a metabolic pathway. They act as a single unit to catalyze a particular reaction. The product of one enzyme is immediately available to the next. An example of a multi-enzyme complex is the complexes within the electron transport chain. Here is the electron transport chain. This is found in the same mitochondrial membrane as the enzyme ATP synthase. The complexes within the electron transport chain are what shuttle the electrons with their hydrogen ions into the outer membrane, creating the hydrogen ion gradient. In step 1 of the electron transport chain, there is a redox reaction of NADH plus H at complex 1. The electrons will go to complex 1, 4 protons pumped from the matrix to the intermembrane space, which is outside of the outer membrane. In step 2, we have the redox reaction of FADH subscript 2 at complex 2. Here's where coenzyme Q, the organic cofactor, picks up electrons from the complexes 1 and 2 and transports them to complex 3. In step 3, the redox of complex 3, 4 protons are pumped from the matrix to the intermembrane space, and a carrier C transports electrons to a complex 4. In step 4, the redox of complex 4, 2 protons are pumped from the matrix to the intermembrane space. We see the formation of water, which forms 20% of the water in our body. And then step 5, ATP synthase action. This pumps protons from the intermembrane space to the interior mitochondrial matrix. This produces the ATP from adenosine diphosphate, inorganic phosphate, and the energy from the redox reactions within the electron transport chain. You do not have to memorize that. This is just showing the complexity of metabolic pathways in the body and how within metabolic pathways we have complexes which are made up of multiple enzymes that all function to do a particular job, such as shuttle hydrogen ions to make the hydrogen ion gradient. Multi-enzyme complexes are regulated through a process called feedback inhibition. Feedback inhibition is a type of negative feedback that occurs when the product increases to a point where it acts as a noncompetitive inhibitor. The product itself will turn off an enzyme located early in the pathway. Once the product has decreased, the feedback inhibition will turn off and the complex will restart. Medicines as well as poisons can act on metabolic pathways by shutting down one enzyme or by shutting down an entire complex. An example of this is cyanide. Cyanide will shut down the electron transport chain by shutting down one of the complexes. By simply shutting down one complex in a metabolic pathway, no ATP can be synthesized. Without ATP, the body cannot perform cellular work. It cannot function, and the organism will die. I'll put the link for the video below. Here is an example of the feedback inhibition. The substrate in red is going to bond to the first enzyme in the metabolic pathway. As the products are formed and they increase, if they get to a certain level, they will bind to an enzyme early in the pathway, in this case enzyme 1. Notice that they are an allosteric noncompetitive inhibitor. They're bonding to a site other than the active site, but they're changing the shape of the active site so that the substrate can no longer bond to it, shutting down the entire pathway. This is important because it ensures that we do not build up too many products within the body. However, it must be able to turn off again when needed in order for the metabolic pathway to initiate once more. This is the illustration of cyanide. It is complex 4 that it binds to. And so if cyanide binds noncompetitively to complex 4, it will alter the shape of its active site. Consequently, electrons cannot be released to oxygen and the electron transport chain shuts down. This is the same comic as prior. Running away from your problems does not count as exercise. Guess it's funny enough, I'll show you again. Thank you for watching and have a blessed day.